Ladies and gentlemen, this video is directed towards anyone who still believes in evolution, atheists, agnostics, or any other group that denies that there is an intelligent designer who created this earth that we live on. It is also directed towards people who already believe in intelligent design. This video should help strengthen your belief. If you didn't see my previous video on the Fibonacci sequence proves intelligent design, then please do if you have the time. It is the same subject matter, so I feel that it will be helpful for you. And just like in that video, this video is not about religion. I'm not going to sit here and push the God did it theory down your throat. That would be an immediate turnoff to evolutionists, and they would probably just stop watching. But what I am going to push, that there is intelligent design behind everything that we see, and then it will be up to you to decide who that creator is. I am going to present solid evidence to you that we did not evolve from a hot, soupy ocean billions and billions of years ago, and that the complexity of DNA, proteins, and the human cells are far greater than anything evolution is capable of. There must have been an intelligent designer who is behind DNA in the human cells, which are supernatural creations. Science just has no valid answer for how DNA in the human cells came to be into existence. Did this exotic car evolve, or did it have a creator, an intelligent designer? Well, that's just stupid. Of course it had a creator. Did this beautiful building evolve, or did it have an intelligent designer? Intelligent designer, of course. What about this jet airplane? Once again, an intelligent designer. Or how about this fine Rolex watch? Evolve or designed? I think you get my point. All these items have intelligence behind them. An engineer, a designer, a creator. So it doesn't matter what group you belong to. We can all agree on this answer. This brings me to the word faith. I hear this all the time. It takes faith to believe in intelligent design. It takes faith to believe there is a creator. Faith here, faith there. Just have faith. No, it doesn't. Not at all. It doesn't take faith to believe that someone designed all these things. But when it comes to the vastly more complex DNA in the human cell, that's right, vastly more complex, which you will see in just a minute, evolutionists immediately say, no, there is no intelligent designer behind these things. They just evolved. No questions asked. In fact, it takes more faith to believe in evolution than it does intelligent design. So why do they do this? This makes no sense to me and has no logic behind it. Maybe they are afraid to admit that there is a creator because then they would have someone to answer to for their behaviors and actions. This may be the case. Just saying. So let's get ready to experience the awe and wonder of how amazingly complex DNA, protein, and the human cell really are. And while you're watching, I want you to ask yourself a few simple questions. Why did evolution give every single one of the 7.5 billion people a unique fingerprint? Why would evolution make it more complex than it had to be? And why would evolution make both male and female? The male and female reproductive parts are very complex, and they are very different. But evolution decided to make both? Why? Why would evolution make it more complex than it had to be? And how would evolution know to make 2 kidneys instead of 1? Where did evolution get its blueprint for this? How did evolution know that humans would be drinking so much liquid over their lifetimes that they would need 2 kidneys to filter the liquid instead of 1? It's these kind of questions that evolutions never seem to ask. Makes me wonder why. So here is the evidence that DNA in the human cell did not evolve over billions of years. And after you watch these, ask yourself this. Since there is an intelligent designer out there, who is it? Who is the creator? Well, you know my answer. It is God of the Bible. So enjoy the videos, and I will see you next time. Let the science lesson begin.
Were we just incredibly lucky? Or was there more involved than blind chance and random interactions of matter and energy? So why is chance such an unsatisfying option? Perhaps because many analysts have done the math and understand the formidable odds against success. Chance is defined as the unknown and unpredictable element in any event that has no assignable cause or guidance from a natural law or intelligent agent. Since the 17th century, one tool has been used consistently to predict the outcome of such events. It is called mathematical probability. In very broad strokes, here's how the theory works. When you flip a coin and call heads, the probability of being correct is 50%, or 1 chance out of 2. Flip 2 coins, and the odds of getting 2 heads simultaneously are 1 chance in 4. 3 coins and 3 heads on a single try, 1 chance in 8. Every time an additional coin is added, the probability of success, in this case all heads on the same toss, is reduced exponentially by a multiple of 2. So if 10 coins are tossed, the probability of 10 heads is 1 in 2 to the 10th power, or less than 1 chance in 1,000. When applied to the origin of life and the random formation of large biomolecules, probability theory clarifies the limitations of chance as a creative agent on the primordial Earth. For example, what are the odds a single protein could form exclusively through the blind interactions of chemistry? Our target is one smaller than average molecule made from 150 amino acids, each aligned to ensure a folded chain. Researchers have calculated that on the ancient Earth, the probability of success was 1 chance in 10 to the 164th power. That's 1 correctly sequenced protein chain for every 100 million trillion 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 failed attempts. But despite these enormous odds, some theorists argue that given enough time, anything is possible. Okay, let's test the validity of this opinion. We'll begin by establishing an ideal environment for chemical evolution. An imaginary world that will provide chance with every opportunity to succeed. First, we stock the oceans to capacity with amino acids. That means all the atoms on Earth, including its entire supply of carbon, nitrogen, oxygen, hydrogen, and sulfur, are available to form 10 to the 41st complete sets of the 20 types of amino acids used to build proteins. Then we'll alter the laws of nature to protect these building blocks from the destructive rays of ultraviolet light and chemical contamination in the primordial soup. Now let's turn the chemistry loose and see what happens. The amino acids start bonding furiously. In our experiment, an entire chain of 150 units self-assembles in only 1 second. Since all 20 types of amino acids are available, at the majority of sites, there is a 5% or 1 in 20 chance the correct molecule will align in the chain. If the sequencing is incorrect, the chain is immediately destroyed and a new assembly begins. Throughout the planet, 6,000 million billion trillion attempts will take place every minute. That means in 4.6 billion years, the oldest estimated age of the Earth, the number of chains that don't fold will exceed 10 to the 58th power. It's a staggering total, but nowhere near 10 to the 164th. The trials necessary, on average, to build a protein of 150 amino acids by chance. So if undirected chemistry can't produce our coveted molecule during the entire history of the Earth, then how much time would have been needed? To find out, let's take a road trip.
Now, suppose against all odds, chemical evolution produced our single functional protein. Would we have life? No. We'd have one protein, just a lifeless arrangement of amino acids. The simplest living cell we know has more than 300 different proteins. But proteins are only part of the story when you consider any actual cell. Remember, you're going to have carbohydrates, complex sugars, nucleic acids, DNA and RNA, lipids, a whole variety of different chemicals which jointly constitute the living state. Those bits and pieces all have to be brought into the same microenvironment at the same moment in time.
Each chemical building block must then be assembled and organized into the network of molecular machines that will control every facet of life.
If we can appreciate exactly how hard it is to produce one molecular machine using nothing except atoms and energy, we can see that there's a profound problem, because once you have one molecular machine, you don't have a living thing. These molecular machines need other molecular machines. And even if nature was capable of producing all the molecular machines necessary, that still wouldn't be enough. They have to all be together, all in this tiny little membrane-bound space that we call a cell.
From my understanding of what it takes to make a cell, it has to happen all at once. You can't do it one bit at a time, because everything works together in a causal loop. The higher level of organization transcends the pieces. The spatial organization in the cell requires that molecules end up in the right place at the right time. The DNA is copied into RNA. The polymerase that does the copying has to find the right spot in the DNA to start copying. The RNA has to somehow hook up with ribosomes, which have to be in a particular place. And the proteins then that are made have to be going to a particular place. That's an awful lot to account for by random chance.
The probability that you would get them in the same space at the same time becomes beyond unimaginable. And the probability that you would get them within a membrane enclosure like a cell is the next best thing to impossible.
Here's how to quickly and definitively refute the theory of evolution. It's simply a fact that life cannot exist without proteins. Proteins are essential to every cell's function and existence. However, proteins require DNA to be formed. That's because proteins are chains of specifically sequenced amino acids. Amino acids must link up in the precise sequence for a protein to form. The precise sequence of amino acids, which is essential to protein formation, is itself determined by the precise arrangement of the bases in the DNA molecule. That means that the precise arrangement of the bases in the DNA molecule constitutes the code for the precise arrangement of amino acids in a chain, and the precise amino acid arrangement in a chain is itself necessary for protein formation. Therefore, to put it simply, to have life, you must have DNA. A protein cannot form without it. But DNA by itself is useless. It can't do anything. DNA doesn't go anywhere or do anything productive without already existing proteins. That's because in order for DNA to be transcribed and utilized in the cell, which is essential to protein formation and life, DNA requires already existing proteins. DNA also cannot even replicate, that is, make a copy of itself, without already existing proteins. Think about that. It means that if you began with DNA but without proteins, you could never get to proteins or life, because DNA needs already existing proteins to even function or form proteins. And you couldn't start with proteins, which are devoid of DNA, because proteins can only be formed from the instructions and information in DNA. That proves that both DNA and proteins, not to mention the many other molecular machines that enable them to interact, must have been present from the beginning of life. One could not have evolved into the other because they require one another for sustenance and utility. They must have been and were created simultaneously. Evolution is false. Charles Darwin, the man who came up with the, quote, theory of evolution, admitted, quote, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down, end quote. We've just discussed a fact which proves, without any doubt, that such a structure exists. There are countless other examples. They are ubiquitous in creation. Evolution is a completely unscientific hoax and fairy tale. It's simply a false religious worldview that has been imposed upon the masses through the education system and by means of lies and hoaxes such as the Piltdown Man. True science, such as the fact that we've just discussed, refutes it, and that is clear to those who actually want the truth. But that's the key, isn't it? Most people don't want the truth. In fact, only a very spiritually darkened generation could have come to embrace evolution and failed to perceive the utter stupidity and illogic inherent in the idea at every turn. And the reason that so many colleges and universities decide to swiftly silence, remove, or intimidate scientists who simply look into the arguments against evolution and begin to speak about them is that the evolution hoax cannot withstand honest scrutiny.
In recent days, the discoveries about DNA in the cell, it has formed a very complex picture. So complex that it has led you to posit that the only place it could come from is from an intelligent designer. Talk a little bit about that and what you're finding in DNA.
Well, as we talked about in our last program, DNA encodes digital information. It's a form of digital nanotechnology. And the information in the DNA directs the construction of the proteins and protein machines that are necessary for the cell to stay alive. And this, I think, has reframed the ancient question about design, because in the 19th century, with Darwin's theory, it was thought that he had explained away all evidence of design. And now we have a striking appearance of design that has no, at this point, naturalistic explanation. And I think there's also a positive reason to think that it was designed by an intelligence. But it's interesting, I think, for our purposes at this point, just to acknowledge that this is a huge mystery. I call it the DNA enigma. Richard Dawkins, who denies that there's any evidence of design, nonetheless has to acknowledge that DNA is full of digital code. He says that the machine code of the genes is uncannily computer-like. Bill Gates goes on to say that DNA is like a software program, only much more complex than any we've ever created. So what we're dealing with inside the cell is information, and that raises a really profound mystery, because we know we need information to build all the important components of the cell. So if you want to explain the origin of the first cell, the origin of life, you have to account for the origin of information. And there's a German scientist named Bernd Olaf Küppers who acknowledged this connection early on. He said that the question of the origin of life is basically equivalent to the question of the origin of biological information. And that makes sense too, because I used to ask my students a question: if you want to give your computer a new function, what do you have to give it? And they would know right away. You got to give it code, information, software. And the same thing is true in life. There's a connection between information and new form and function. And if you want to get a functional living organism off the ground, you got to have that information. So that's the DNA enigma. That's the big mystery that has to be solved.
And it's a lot of information. It's not just a few things. It's a lot of things in a lot of different parts that are all programmed to work together. It's very complex.
It's lines and lines of code. I mean, in the human genome, you've got 3 billion characters. In lower animals, somewhat less, but it's an amazing amount of information in every living organism.
Now we're going to show you a little bit of this complexity, and the big question that we're going to is, where did this information come from? If you're starting from zero, could this information have come about by chance? Natural selection. All right, so let's watch this animation, and I want you to see this. Watch.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a 2-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
All right, folks, that's just one cell. All those little parts are in just one cell, and information is king. Information is controlling all of those different parts. It's even more complex than what Stephen just showed you here. Stephen, where did this information come from?
Well, that's the $64,000 question, as they used to say. It's I call it the DNA enigma, and the DNA enigma is not what is the structure of DNA. Watson and Crick solve that mystery. It's not even what does the information do. We've just seen it in the animation. The question is, where did the information come from? And there's 2 basic answers. Either the information arose through purely undirected, unguided, purposeless material processes, or it's the product of some kind of designing intelligence, in what we call intelligent design. Those were the 2 options, and that's what I set out to investigate as I became fascinated with this question in the mid-1980s.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
One machine particularly attracted his attention.
I remember the first time I looked in a biochemistry textbook and I saw a drawing of something called a bacterial flagellum with all of its parts in all of its glory. It had a propeller and the hook region and the drive shaft and the motor and so on. I looked at that and I said, that's an outboard motor. That's designed, that's no chance assemblage of parts.
Behe's reaction was not surprising, for the molecular motors that drive bacteria through liquid each depend upon a system of intricately arranged mechanical parts. These parts come into focus when portions of a cell are magnified 50,000 times. Biochemists have used electron micrographs like this one to identify the parts and three-dimensional structure of the flagellar motor. In the process, they have revealed a marvel of engineering on a miniaturized scale.
Howard Berg at Harvard has labeled it the most efficient machine in the universe. These machines, some of them are running at 100,000 RPMs and are hardwired into a signal transduction or sensory mechanism so that it's getting feedback from the environment.
And even though they're spinning that fast, they can stop on a dime. It only takes a quarter turn for them to stop and shift directions and start spinning 100,000 RPM in the other direction.
And just like outboard motors on motorboats, it has a large number of parts which are necessary for the motor to work.
The bacterial flagellum, 2 gears, forward and reverse, water-cooled, proton motive force. It has a stator, it has a rotor, it has a U-joint, it has a drive shaft, it has a propeller, and they function as these parts of machines. It's not convenient that we give them these names. That's truly their function.
Since its discovery, scientists have tried to understand how a rotary motor could have arisen through natural selection. As yet, they have failed to offer any detailed Darwinian explanation. To see why, we must understand a feature of molecular machines known as irreducible complexity.
Irreducible complexity was coined by Mike Behe in describing these molecular machines. Basically what it says is that you have multi-component parts to any given organelle or system in a cell, all of which are necessary for function. That is, if you remove one part, you lose function of that system.
The idea of irreducible complexity can be illustrated by a familiar, non-biological machine, a mousetrap. The trap is composed of 5 basic pieces: a catch to hold the bait, a strong spring, a thin bent rod called the hammer, a holding bar to secure the hammer in place, and a platform upon which the entire system is mounted. If any one of these parts is missing or defective, the mechanism will not work. All components of this irreducibly complex system must be present simultaneously for the machine to perform its function, catching mice. Irreducible complexity also applies to biological machines, including the bacterial flagellar motor.
All told, there are about 40 different protein parts which are necessary for this machine to work. And if any of those parts are missing, then either you get a flagellum that doesn't work because it's missing the hook or it's missing the drive shaft or whatever, or it doesn't even get built within the cell.
In evolutionary terms, you have to be able to explain how you can build this system gradually when there's no function until you have all those parts in place.
It wasn't until 1839 that cell theory was first described by Theodor Schwann in basically its modern form. By 1858, researcher Rudolf Virchow had learned enough about the cell to conclude that every cell must come from a pre-existing cell. But Charles Darwin wasn't paying attention. The very next year he published his book on the origin of species, in which he theorized that the first cell was formed from non-living matter. In Darwin's day, the cell was thought to be just a simple sac filled with jellied carbon. This concept is the origin of the term protoplasm. However, nearly 150 years of cell research has shown us that even protozoans and fungi cells are hugely complex. Many thousands of different operations are taking place continuously within each of the cell's many parts, called organelles. The cell cannot live until all these parts are working properly. Even the simple E. coli bacterium has 4,000 genes. If we were able to magnify the DNA of the E. coli to the thickness of a clothesline, it would be 5 miles long. There is no such thing as a simple cell. The fact that a cell cannot live without all these thousands of parts shows that the cells were created in their finished form, just as the Bible says. Even bacteria declare the glory of our Creator.
And that is DNA, or biological information. In fact, scientists, I'm sure you read about this not long ago, finished mapping the human genome. And it was very interesting. The human genome is the blueprint inside every cell that explains how to build all the proteins in your body. And I love the words of President Clinton when they announced this breakthrough. They'd actually mapped the human genome. He said, "Today, we are learning the language in which God created life." And that's exactly true. DNA is quite literally language. It's language. Inside every one of your 100 trillion cells in your body is this famous double helix of DNA. And if you were to open it up and stretch it out, it would stretch out 6 feet long inside every one of your cells. And it is encoded with a 4-character chemical alphabet that precisely spells out the assembly instructions for all of the thousands of different kinds of proteins out of which your body is constructed. In fact, DNA is the most efficient information storage system in the universe. Our science cannot come anywhere near replicating the ability of DNA to contain information. I'll give you an example. This will blow your mind. Picture a teaspoon of peanut butter. You know what that looks like? A teaspoon of peanut. Now imagine that's not peanut butter. Imagine that is pure DNA. How much genetic information could that teaspoon of DNA hold? Just one teaspoon of DNA could hold all of the precise assembly instructions for all of the proteins of every one of the 1,000 million species of animals that have ever existed on the planet, and still have information left over for all of the information in every book ever written. That is how efficient DNA is in storing information.
If simple water molecules that form ice crystals exhibit magnificent structure, consider the design ingenuity behind large, complex molecules, such as DNA. DNA contains the blueprint for all life and is by far the densest information storage mechanism known in the universe. For example, the amount of information contained in a pinhead volume of DNA would fill a stack of books 500 times higher than from here to the moon. The program code and design of such an incredible system indicates a supremely intelligent designer.
The evidence to me that just cries out that there's a God is the study of DNA. DNA is a very powerful, massive information storage system. In fact, DNA that makes up our genes actually is like books of information that's read by a language system. It's absolutely phenomenal. And scientists know today that languages, a code, only come from an intelligence, and information only comes from information. Nobody's ever seen matter by itself give rise to a code. Nobody's ever seen matter by itself give rise to information. And as you look at DNA, it actually cries out, in the beginning, God created the universe.
We all begin as a single cell the size of a period at the end of a sentence. How does that cell know how to build a body with 100 trillion cells in it, thousands of different kinds, and each one of them is so complex, nanochemical machinery beyond our comprehension how it works, and encoded is the instruction manual. It's the manufacturer's manual how to build and operate every part of this incredible body made up of 100 trillion cells.
Furthermore, DNA is a three-dimensional molecule that is self-replicating. Each molecule is able to make an identical copy quickly and efficiently. The Lord has even programmed DNA to detect and correct replication errors. These sophisticated capabilities far exceed man's means.
God has created the DNA molecule in such a way that it is self-correcting. There are special proteins called enzymes that go up and down the DNA molecule looking for and making repairs on a minute-by-minute, second-by-second basis. God created us with a DNA code that actually has what we call editase or editorial type enzymes. Just as an editor reads a newspaper or a book looking for mistakes, so God has created special enzymes that go up and down our DNA molecule, repairing the mistakes in ways that are unbelievably complex.
There are many examples in creation of things that demonstrate the biblical God. One would be in our very DNA. Our DNA has information in it. And there is a whole field of scientific study called information science, which studies how information originates, how it's transmitted, and so on. And one of the laws of information science says that information never originates by itself in matter, never spontaneously comes about. Anytime we trace the copying of information back to its source, it always comes back to a mind. And since we have creative information in DNA, that tells me that DNA comes from intelligence. It's not something that could possibly come about through millions of years of mutations and natural selection. That just won't work.
Yet even the DNA molecule is simple compared to cells. All life consists of cells, and each cell functions as a miniature city. When we consider that a human body consists of trillions of cells working together as one unit, we should be in humble awe of our Creator's intimate care and perfect wisdom.
Scientists generally consider anything with a probability of less than 1 part in 10 to the 70th power operationally impossible. And by the way, that calculation doesn't take into consideration the information that's stored in the cell, which directs the cell in all aspects of operation. That estimate is solely the chance of chemicals combining to form something living. So, the chance of life evolving and retaining the information needed to replicate itself is astronomically smaller than anything we know. And that's only one organism. Think about what it would mean for millions of complex organisms to have evolved from an undirected natural process. According to information science, the probability is so small that it's deemed operationally impossible. So, knowing all of this, should the popular scenarios of chemical and biological evolution be taught globally as the only explanation of the origin of life and species? It's my belief that we're not opening our minds to the possibility of other explanations. Now, one of the most basic concepts you should have learned is that if observations and data contradict the theory you're testing, then the theory should be modified or abandoned. Unfortunately, this doesn't seem to be happening to the present, most popular model of origins. Instead, many scientists are trying to take information and make it fit the evolutionary models. But is that good science?
Critical thinking is required to realize the information science aspects of this and to make sure that whatever scenarios you're coming up with do not violate the principles of information science. Too often, scientists, they believe that information can be generated by physical processes, and that simply is not true. Functional information cannot be generated from purely physical properties.
Sigmund Freud once said, "From error to error, one discovers the entire truth." As we examine history, we are constantly reminded of our ever-evolving thoughts in science. And in the case of the cell, the more we research, the more complex it seems to be. As we gather information, it is up to us as scientists, students, and colleagues to bring science to a level of integrity and critical examination that it deserves. If we approach science with an unsupported prearranged bias, then what we're trying to accomplish is not really science at all. The beauty of science is that we're able to move away from accepted dogma to examine the evidences. It's not up to us to disprove a given theory. It's up to the theory to prove itself against the laws of science. If a theory fails to do this, then it should be rejected, and we should search for more knowledge in order to, as Sigmund Freud said, discover the entire truth. The possibility of life evolving using the known laws of chemistry and physics is operationally impossible. When we consider the laws of information, that possibility now becomes impossible. Meaningful prescriptive information cannot arise from nothing, no matter how much time you allow. And until we acknowledge this, we will never discover the origin of life.